Hello everybody and welcome to today's webinar. My name's Melissa and I'm your host for today in what is our last webinar in our series, Bringing Data to Life, Data Management for the Biomolecular Sciences. So now I'm going to introduce the speakers for today. So we have two speakers today. So we have myself and Sarah Morgan. Sarah is the Scientific Training Program Coordinator at EMBL-EBI. Sarah has a Bachelor of Science and an MSc in Biomedical Sciences, followed by a PhD from Cranfield University and later a Master of Arts in Healthcare and Law and Ethics. Over a 10-year academic career, her research has focused on tumour biomarker characterization and cell surface interaction. Beyond research, she took an active role in the provision of postgraduate taught courses for the university as a lecturer in molecular medicine, leading the development and direction of a number of Master of Science courses, including the Master of Science in Translational Medicine, the Master of Science in Molecular Medicine, and more recently acting as a director for the Master of Science programs in Advanced Biosciences. She has lectured on a variety of biomedical techniques and topics, including ethics and governance of research. So this is where I have the unusual pleasure of introducing myself. So I'm a Scientific Training Officer in e-learning at EMBL-EBI. So my role involves organising online courses such as this one. I joined the training team in 2016, having been a curator for Array Express here at EMBL-EBI. And before that, I was a postdoc where I worked on infectious diseases in the lab. So with that, I'm going to switch off my webcam and I'm going to hand over to Sarah, who will get us started with the webinar.
Okay, so thank you, Melissa, for that introduction, and a welcome from me to today's webinar as well. So this is the last webinar in our series around data management, and the reason for this one really was to try and summarise some of the questions, some of the myths that have arisen, and also to look at some of the top tips and guidance that a number of our trainers have delivered over the series of webinars we've run. So really it's trying to look in a little more detail at the questions and try and summarise everything for you. Now, hopefully a number of you have already seen the webinars we've run so far in this series. Just launched a quick poll to ask you how many of the others you've seen. But through the series, we've gone through a variety, from starting with, what is data management, looking at ways of being able to plan your data management, looking at tools for doing so, and looking at issues around open data, sharing data, why you should do it, what's in it for you, how to do it. And really what we want to do in the webinar today, as I say, is look at some of these issues in a little more detail. During the webinars, we've had a number of fantastic questions raised. So we wanted to bring some of those out again, explore them in a little more detail, and also, as I said, provide a nice summary of the tips and best practice that has been suggested by the trainers as we've gone through the series. So I guess the first thing really is to look at, what do we actually mean by data management? Oh, sorry, quick result there. So you've all watched other webinars in the series, so that's great. So hopefully, as I say, this will be a reminder of some of the things you've seen, but also maybe give you a chance to, if you didn't watch all of them, pull out some of the things you may have missed. And as Melissa said, all of the webinars are there for you to watch if you did miss any of the previous ones. So starting today, really it's sort of looking at what do we mean by data management. And data management, as you can see here, really is a whole cycle around research data. Everything to do with planning and designing what your data, what you're actually going to be collecting, how you're collecting it, analysing it, and also what you do there afterwards. So, do you put it out there in a publication? Do you share it with others? Are others then reusing your material? And this is what we've been talking about really throughout the series, and this applies whether you're talking a small project, which it's just you are doing, for example, as part of a PhD or part of a program of study, or whether you're part of a large consortium where you've got huge amounts of data being collected by a number of different organisations, a number of different groups. The same principles apply. So why data management? These are some of the questions we've had that have been raised. You know, why do you need to worry about data management? Whose job is it? Is it just something you need to worry about if you're talking large data, or if, you know, does it matter if you're talking small data? So Melissa, what do we need? Why do we need to think about this? Why do we need to worry?
So I think we've heard a few different reasons during the course of the webinar series. And the first one is that a lot of the key decision makers, like the funders and the journals, are now requiring you to have a data management plan, and they're rewarding people who do have data management plans as part of their grant applications. So for example, the European Commission is now looking at ways to reward people who are really engaging with and getting involved in open science practices. But of course, it's not just all about the funders and the journals. There are lots of reasons and things that will benefit you in having a good data management practices in place as well. So for example, as Rob mentioned last week, we are the first reusers of our own data. So having a plan and having your data well organized makes accessing and using it much easier. As Elizabeth pointed out in our second webinar, it's also a way for you to give back to the community. So if you're using data from different databases, by sharing your data, it's a way to help build those resources as well. Having good data management practices in place is also really good for your reputation. It helps make your work more visible, more reproducible, and therefore more citable. So you end up having more collaborations and working relationships with other people. And it shows them that you know what you're doing, and that it gives you the chance to think through what you're doing as well. And last but not least, you'll get nice emails thanking you for having done a good job. And I liked Elizabeth's example here, where she said that you can become the smug data angel by getting these nice emails.
So hopefully, you know, you're convinced at this point that, you know, thinking about data management is important to you, not just to others. But the first question really is, how do you manage your data? So, you know, where do you start? What do you do? The first thing is make a plan. So Melissa, tell us a little about making a plan.
Yeah, so making a plan, I think, is the most important thing. And again, Rob said last week, he used this quote from Eisenhower, that in preparing for battle, I've always found that plans are useless, but planning is indispensable. And it's really the same with your data. It's the process of making that plan and sitting down and thinking about what you're going to do and how you're going to do it. And through doing that, discovering things that you might not have thought about, that is actually the most important thing. I know for many people, if you've never made a plan before, this can be quite daunting and you don't quite know where to start. So I think when you are making a data management plan, the thing to keep in mind is that data management life cycle. So you need to be thinking about all the things that make up that life cycle, how you're going to collect and document your data, how you're going to analyze it, store it, and share it. But you don't need to do this on your own either. There's lots of online tools that are already there. For example, we heard about DMP Online last week and the Data Stewardship Wizard, which are both online tools that can walk you through the process step by step. The other tip here is just to go and talk to people in your own institute and see what they're doing as well. There's usually somebody around who can help you do this.
So, you know, you need to make your plan. The first step in terms of that plan really should be keeping good records and making sure that you've got a really good track of you know, everything you're doing, everything you've done. We've got a quick photo of you here and a quick question really. Do you think that either of these looks like good data management? So what are your thoughts? I must admit that having worked for a number of years in university labs, the picture on the right where people have scribbled on it on a piece of tissue paper, I have seen quite a lot.
I have to confess that's a picture of my desk.
Melissa.
And also my freezer did look quite a lot like that as well. Okay, so I'm going to close that poll now.
No, excellent. See, you all win gold stars for this, definitely. This is not great data management. We know that by looking at it. But to be fair, trying to think about how you can actually record your data in a useful manner can sometimes be difficult. So what are we talking about? What kind of data? How do we start?
Okay, so what kind of data? So actually, it's not just about the data. So some things to remember when you are collecting the data is a good way to think about it, I guess, is that you need to collect enough information so that you, as your primary collaborator, can come back in 6 months' time and still understand what you did. So this is a quote from a paper that I read a little while ago on data management. And the quote was, you are your primary collaborator. I can't quite read it. Let me just minimize this thing on my screen. So your primary collaborator is yourself 6 months from now, and your past self doesn't answer emails. The other thing you might want to keep in mind when you do start collecting your data are the FAIR data management principles. So these describe the ways that you can make your data findable, accessible, interoperable, and reproducible. And you can learn all about them in the paper that I've linked on this slide.
So as Melissa just said, it's not just about the actual kind of data that you're generating, but it is also thinking about the other information alongside that data that makes it more useful to use. And this is the kind of thing that people often forget to link together when they're trying to record all this information. So Melissa's going to take you through a little bit more detail about this now.
So as I started to say earlier, it's not just about the data. You also need to collect metadata, which is information about the data that gives it context. And it's the minimal information required to unambiguously describe the experiment. So this includes things like the experimental background, what the aim of the experiment was, who did it, and what the main variables were, the types of samples that you had, and the protocols that you used, both in the wet lab and in the dry lab, so when you're analyzing your data, the raw files and the processed data files as well. Luckily, there are lots of standards in place that help you understand what it is you need to collect. And these standards will describe what that minimal information is, but they also tell you about the format that you need to collect your data in. For example, for RNA-seq experiments, the standard is called MinSeqi, and it describes all of these things that you need to collect. And you can learn more about the different standards available on fairsharing.org, and there are many different standards available for different types of data. So for example, for microarray data, it's called MyEMI. For metabolomics, it's MSI, and proteomics is MyAPI. So a good tip here is to go and learn about the standards that are being used in your field.
So one of the issues in terms of trying to record things and thinking back to the image we showed you just then with your your variety of tubes just sitting in a tube rack, one of the challenges that we often get asked about is, how do you label things, especially if you're part of a wider group, a team effort? How do you actually go around labeling things in a way that somebody understands?
So labeling things is always something that I was good at, and then I would go back to my lab book and I'd have no idea what I'd written down. So I think it's quite a common problem. And we can see that here, that sometimes you need to be careful to keep things consistent, both within your own experiments, across experiments you're doing within your lab, and then further afield, even with the field in general as well. So in this example, there are lots of inconsistencies. For example, the way that the samples are named is not consistent, and the way that the files is named is inconsistent in both the format and the way that the samples and the file names relate to each other. Similarly, there are several different words used to describe a treated sample. For example, we've got treated, isoniazid, INH, and the chemical formula for isoniazid on the bottom there. They're all exactly the same thing. So when you are designing your experiment, sit down with everybody who's involved and agree on a way to label your different samples and agree on the words that you're going to use to describe them. So this is the same data set, and you can see straight away by having a consistent way of labeling things, it's much easier, at least visually, to see what went on in an experiment. It's much clearer that there are 4 control samples and 4 treated samples. It's much clearer to see which file belongs to which sample as well. So one of the ways that you can do this, because we're well aware that as humans we have many words for the same thing. So one of the tools that you can use is an ontology. So an ontology is an agreed vocabulary or a way of describing things, for example, experimental vocabularies. And the cool thing about ontologies is that within the ontology, there is information about how the terms relate to each other and the synonyms of the terms as well. And this becomes really useful when you need to search for information within a database, for example, because by using an ontology, if your database is using an ontology, it knows that if you search for human, for example, you also mean to search for man and Homo sapiens. Similarly, if you're searching for brain, it knows that you also want to bring back results that have the word cerebrum or cerebellum, because it knows those two things are part of the brain. So the plus side for you is if you're using an ontology to annotate your data, it makes it much more discoverable because people don't have to do as many different searches to bring back those results.
So ontology is obviously really helpful, as you're saying, in terms of being able to annotate so you can find things, but there are so many ontologies out there. How do you decide, or where do you find information on the ontologies that are there, and which one is best to use for your circumstance?
So I'm not sure about deciding which is the best one to use for your circumstance. I think that's up for you and your team to decide and to discuss. But if you're looking for a particular term or you want to check something, we have a tool called the Ontology Lookup Service at the EBI, where you can go to it, you can type in a particular word, for example, cerebrum, and it will tell you what the appropriate ontology term is for that word. As Sarah alluded to, there's lots of different types of ontologies in biology. One that we use a lot here at the EBI is the experimental factor ontology, which is used to describe all the different variables that you have in an experiment. There's also the gene ontology, which quite a lot of people have heard of. So that describes what genes do, where they are within a cell, and their molecular function. And there's also the plant environment ontology, which is an agreed way of describing the conditions in which plants were grown. So I just wanted to add this on the end. So it's just one more thing to look out for in terms of being consistent with your data. And we're all human, we all make mistakes, and you know that sometimes you just can't see it for looking. So just, it's really good if you just take a moment just to double check your data, get somebody to have a look at it as well, and make sure that it makes sense. Make sure that you haven't accidentally labeled your control sample as having been treated and vice versa. When I was a curator, it was surprising how often these things would come through, and even as a curator, you would make the same mistakes as well, and then another curator would pick it up.
So having thought about, you've generated this data, it's recorded, you've got it lovely and organized, you now want to start sharing it. And this is obviously something we touched upon in one of the webinars, looking in more detail at sharing. And during the webinar we ran around this, we actually asked people whether or not they shared their data. The nice thing was to see that a number of people had, but it was interesting to see that a number of people had not, but would like to. And what we wondered was really what was kind of holding them back. Where were the issues? Where were the challenges, the concerns, etc. in doing so? So we had a bit of a look at this, and this is where we've got a whole host of different comments that people come back with. People querying whether they can share data that isn't published. Will they be scooped if it gets shared before publication? How are they going to benefit? Will they benefit from sharing data? And issues around sort of trusting other scientists to cite their data when they're publishing things, and also, okay, my data, it's too big, my file's too big. Well, how do I deal with this? So this is something we had a bit of a look at. And what we're going to take you through now is a few things that you need to know about sharing data and how you won't need to worry about the issues that many people have raised.
So I think a lot of these issues they're completely understandable. They're things that I've come across as a researcher as well. On the topic of benefiting from sharing, it's actually quite similar to the benefits that you get from having good data management practices. And getting your data out there helps build your reputation and helps other people find you, to cite you, and to collaborate you. Some other things that you need to know about sharing, and many people don't know about sharing, for example, are that the data can be kept private until it is published or until you agree for it to be released. So for example, this is the sort of timeline of data submission and what happens to it in one of our databases here called metabolites. So you can see that when you first submit your data, only you and the database, the people who work from the database can see it. Then while it's being curated, it's the curators and you who can see it. After that, they can give you a link that you can share with reviewers so that they can see it during the journal review process, but it is still not public for everybody to see. And once you have decided to make it public, then everybody can see it. So you can still, this is one way that you can protect yourself from being scooped. Although I have to say that I've never actually seen anybody scoop somebody's data out of a database and publish it as their own. The next things to note are we have ways to handle big data. So at the EBI, well, we specialize in handling data and in sharing it with other people. And there are ways that we can upload very large data sets, for example, across our FTP server or using tools like Aspera. Or in some cases, we also sent hard drives to people and had them FedEx them back to us. So that's always an option as well. However, if it really is impossible to share your data because of the size of it, for example, at least share the metadata so that your story and your data and your work is still discoverable. You can still benefit from sharing the metadata. So I'm going to hand back over to Sarah now because she's the ethics and clinical data expert between the two of us, and she's going to tell us a little bit about specialized databases for human data.
So one of the big areas of concern that we've again had raised by a number of people is around where you've got data, where you've got the medical information alongside the experimental data you've generated. Now, obviously, this is a huge concern because the medical data is highly confidential, is going to, you know, there's a lot of personal information in there. So how do you deal with that? Obviously, a number of places, such as biobanks and things like this, where they're collecting this data, they will have various levels of access. So, you know, they will anonymize certain sets of data to hand out. However, of course, for many instances, it's actually useful to have that medical data alongside the experimental data so that you can get a fuller picture of what's happening. This is where databases such as the European Genome Phenome Archive come in. So the way the EGA works is that it does contain both genome data and the medical data. However, there are data access committees in place. So the people who submitted that original data still get to look at anybody who requests access. So it's not fully open access, as in you can just go and search through the database. But if there is a specific data set in there that is useful to your research, you can request access. And as long as you have a good reason for accessing that, and a very clear reason, a very clear piece of research, the likelihood is you will be granted access to it. So it's just that extra step to make sure that, you know, people aren't just reviewing it, they're actually using it, and what reasons are they using it for. So, you know, there are more and more databases of clinical data out there. There's more and more of a need for this to happen. And as I say, it's, you know, don't be put off by the fact that you've got that kind of data. There will be a way of being able to share the information so that you're not causing any ethical concerns for those patients and others who gave you that sample in the first place. So the next thing, though, from, you know, obviously we've talked a little bit there about repositories, you know, where do you deposit your data? Because obviously, you know, again, there's a huge choice potentially in databases where you can deposit this data, but how do you go about choosing the best place to go?
Mm-hmm. So there are many databases in place for many different types of data, and it's true, the array of different databases can be a bit overwhelming. Luckily, there are databases about the databases where you can go and look these things up. So two that we've come across during this series are the r3data.org and fairsharing.org. So those are two places online where you can go to look for a database that suits your particular type of data. It's also a good idea to check in with the journals because often they'll recommend particular databases. And you can also talk to other people as well, see what they're doing. This is particularly useful if you're in a new field and the kind of hasn't quite been worked out yet what the best place or way to store your data is. Now, if you want to submit your data to the EBI and you're not sure of which EBI database to use, we have a tool that can help you figure that out. So if you go to our homepage, which is at www.ebi.ac.uk, you'll find this deposit data button. And if you click on that, it will take you through to our data submission wizard, which will ask you a short series of questions about the type of data that you have and what you want to do with it. And it will suggest a database that's suitable to your needs. And once it has suggested that, you can link through to that database so that you can get in touch with them and ask them questions about what you need to do to submit your data there.
So during one of our webinars, we actually had a number of curators online talking about data sharing and looking at it and from their point of view. And actually, as part of this process of devising these webinars generally, we spoke to curators. We have a lot of curators at the EBI who work with researchers to bring their data into the databases. And we asked them what were their top tips for sharing data.
Yeah, so our curators' top tips for sharing data are to start early. Because you can keep your data private until you're ready to release it, you can start early and start collecting that metadata as you go along. This makes the process of submitting it and getting your stable accession number much quicker. The second tip is to get in touch with them and talk to the curators. The curators are your friends. It's their job to help you get your data into their database, and they do it all of the time. They've seen every type of data under the sun. They also really recommend that you do find out what the requirements are and what the standards are in terms of the file formats, etc., for the particular type of data that you are collecting. And again, just remember that your data can be kept private until it's published or you ask for it to be released, whichever occurs first.
So having shared your data, the next area of concern, and this came up when we actually did a brief survey when we were designing this webinar series, was around citing data, whether it be how to do it or actually how you can make sure that people cite the data that you've generated and have shared. So one of the other, the one question that really comes up is how can we actually trust other scientists to cite us? So the example we have here is someone who's generated some genome data. If we share this before we publish the related paper, how do we know, we can actually trust somebody not to do something with that before we do? And also, the idea of, well, if I'm citing a publication where they talk about the data that's been generated, do I actually need to cite the data set as well? So, Melissa.
So I think the way we think about what we cite has changed over time. It used to be that the journals and the articles that we're publishing were the center of the story, but increasingly it's the data that's the center of that story. So I think it's actually really important to cite the data as well as the paper, particularly because the paper might tell part of the story, but not all of it. In terms of how can we trust other scientists to cite us, I think on some level we just have to trust them, wouldn't you say?
Yeah, I would agree.
Yeah.
Yeah.
But you can also make it easier for them to do it as well. So for example, you can, when you do submit your data to a database, you're given an accession number or a DOI. So when you do write your paper, make sure that you're including this accession number and the DOI, and you cite the database in your article. This makes it much easier for somebody else who wants to use your data to make sure that they're citing you. Make yourself easy to find as well. So you can use your ORCID ID to do this. An ORCID ID is a unique identifier for a researcher, and it follows you across the length of your career, regardless of name changes or changes in where you work, etc. So this makes it easy for people to get in touch with you so that they can ask you about your data. And if they've had personal contact with you, they're far more likely to cite you. An approach that Elizabeth Bush took in their pre-publication data, which appears in Array Express and Expression Atlas, was to put a disclaimer on their data, asking people to refrain from doing a full analysis until they were able to publish it themselves. but saying that equally people could use it in parts, so long as they cite it and that they cite it correctly.
So there are a few of the questions that came through as around data ownership. It's obviously an area of concern for a huge number of people. So one of the first questions really was, when you actually submit your data, so if I've submitted my data, who actually owns it once it's been submitted?
So from the database perspective, the ownership stays with the submitter. And if that data is reused, it needs to be cited, and so that it can be linked back to the person who submitted it. In terms of who owns it within your institute, that's probably dependent on the agreement that you have with your institute.
Yeah, definitely. How about linking data sets to my ORCID ID?
Yes, as I was just saying, you can do that now, and some databases will allow you to do that automatically. So either you can supply your ORCID ID when you submit your data, or they have a claiming tool as well.
And what about linking to publications or pre-publication things in bioarchive and this kind of thing? Is that still possible?
Yes, no problem with that at all. The same way that you would do in a reviewed article, you just put the accession number in and the name of the database.
And finally, I guess, we've got more and more scientists we see them now coming through to training where you've got multi-omic data sets. Now, obviously, those sets of data, whether, proteomics is going to sit in one database, the genomic data will sit somewhere else. Is there any way of actually being able to link those data sets together?
Yes, there are ways to do that. For example, when I worked for Array Express, we sometimes saw data sets come in where they'd have, for example, RNA-seq data as well as proteomics data. And what we would do is we would, in the metadata we were collecting, so the description of that study, we would write down that this has a linked data set in PRIDE, the protein database, and the accession number is. So it's just a matter of being a bit proactive about that and making sure that you're making the effort to link those two together.
Excellent. So thinking now as a bit of a summary really of all of these issues around data management, what's the big take-home message, I guess? One of the big things is ask for help. If you don't know what to do, ask for help. And what we want to show you now is just a few areas of where you can find some more of this help, aside from this webinar series, of course.
Yeah, so asking for help is my bioinformatics superpower. I think this is one of the most powerful things that you can do. So in terms of places to find help, of course, we've mentioned a few as we've moved through this webinar series, and you'll find more links in the recorded versions. So fairsharing.org is a great resource. It will help you learn about all the different standards and the FAIR principles, and it can also be used to find databases. Again, once you've found those databases, speak to the curators and the people who work for them. They're the experts, they know what they're doing, and it's their job to help you. Equally, speak to the people who are working around you and check to see if there's any support available through your own institution. Increasingly, many universities and research institutions have data management resources and people whose job it is to help you with your data management. Another online resource is the Digital Curation Centre, and although it is slightly UK-centric, it has a lot of information in there that's applicable across Europe, the US, and beyond as well. And of course, as we've plugged a couple of times already, you can always rewatch these webinars in Train Online, and there are extra links and hints and tips in there as well. So this brings us to the end of the webinar. We wanted to give you a few dot points or bullet points, as Sarah, we sometimes have a discussion about which is the right term. We wanted to give you a few take-home points, really. So the main things for you to think about in terms of data management for your own research are to start early. Make a plan and keep good records, and in doing so, make sure that you're also collecting metadata, that you learn about the standards, that you're consistent in the way that you are annotating and speaking about your data, and be careful of human error. When you do come to publishing your data, make sure that you're including the accession number and the name of the database. And equally, if you're using somebody else's data, please make sure to cite them. Data is what we're creating. It's what our careers are now built on. So make sure that you're being fair to your other researchers as well. Make them feel as though they can trust you, I think. And last but not least, be brave and ask for help. So that brings us to the end. We do have time for questions. If you would like to ask us a question, please do write it into the chat box and we will read them out and answer them the best that we can.
And whilst you're thinking of your questions, we'd just like to give a quick thank you really to everybody who has helped with this series and get this off the ground. We've had a huge number of speakers involved, but also, as you can see from the lovely shot of the whiteboard we've got here, we had a fantastic brainstorm session with a number of trainers from across the EBI and beyond to think about what we should include in this. Everything is up there on Train Online for you to go and have a look at. We hope to, or we will be adding in more information actually as we go through as well. So it will build into a slightly larger resource. So again, if there are other things you think would be nice to have in there, please do tell us. But as Melissa said, we're here for a few more moments to answer any questions you may well have. So please ask away.
Okay, so there don't seem to be any questions today. So just one last thing to wrap up. Again, you can find the full series in Train Online by following this link. And please don't forget to fill in the feedback survey, which will pop up in your window once we close this webinar. Just to follow on from Sarah's thanks, thank you to all of the speakers and the organising committee who helped us create this webinar, and also to everybody who's joined us and tuned in. We couldn't have done it without you, and we hope to see you again in future webinars. Thank you and enjoy the rest of your day.